Hello, my name is Felipe Paes Gomes da Silva and I'm going to present my work entitled Pulmonar expression of angiopoetin-1 in patients who died from SARS-CoV-2 and H1N1 pandemic viral pneumonia. But actually we have to make some changes in the process, and we study now the molecular epoxy-inducible factor 1 alpha. So here we go. When the SARS-CoV-2 gets in the respiratory tract, it triggers a cytokine storm that promotes structural and metabolic changes. The focus of our study was to see this cytokine storm caused by SARS-CoV-2 in the pulmonary vascular endothelium, and we can saw some microthrombosis events in vascular remodeling and also angiogenesis process. The process of angiogenesis comprises the formation of new vessels, and this could be physiological and not physiological. When not physiological, these vessels can grow like false bottom or fake vessels and promote the microthrombosis. Some studies already done with patients who died from COVID-19 and had angiogenesis process involved showed for us that this factor is involved in the process. And in the presence of oxygen, the HIF-alpha is degraded. The objective of our study was to evaluate the tissue expression of HIF-alpha in the lung samples of the group SARS-CoV-2 and H1N1 and the group control. Also, we correlated tissue expression of HIF-alpha with time of mechanical ventilation, and also we suggest a possible molecular pathway of angiogenesis in COVID-19 group. So our monitoring methods comprise post-mortem lung biopsies of 3 groups: control, COVID-19, H1N1. The N of individuals is shown in the picture. We took clinical data from the patients who died from COVID-19. This clinical data is the time of mechanical ventilation. All the samples from the lung biopsies moved to histopathological and immunohistochemistry analysis. And then the software amplified the image of the samples and sent to statistical analysis. Our results, the analysis of the tissue expression of the HIF-alpha was shown to be as higher as in the other groups. We didn't remove statistical analysis and difference, but when compared the HIF-alpha with time of mechanical ventilation in days, in COVID-19 and H1N1 group, we saw that the H1N1 group, who stayed less time on mechanical ventilation, had more expression of HIF-alpha, and it suggests that HIF-alpha is more present and more activated in the epoxy environment. And also we suggested the pathway of angiogenesis. And in response to entry of the virus, macrophages of M1 phenotype secretes pro-inflammatory cytokines, such as TNF-alpha, which binds to TNF-1 receptors. As a result, there is an intracellular sign response which releases the NFKB from his invader, allowing the NFKB to be activated and enter the nucleus for its transcription action. The NFKB acts by binding to the initiation site of HIF-1 precursor DNA, which would be translated in protein form. In contact with the epoxy environment, HIF-alpha is not degraded. It's able to enter the nucleus to act on the transcription of the RNA of the vascular endothelium growth factor, which promotes the angiogenesis process. And here's my publications during the time of this project. And we are waiting for the journal answer for this new publication. My reference are in the correct code.